Welcome everyone to this new Genetic Engineering and Biotechnology News webinar sponsored by GenScript entitled Multidimensional Approach to Decoding the Mysteries of Animal Development. I'm Jeff Bogaliscus. I'll be your host today. I'm the technical editor for Gen, which is rapidly approaching its 40th year covering all aspects of the life science industry. Today we're going to be discussing molecular regulatory elements that control development and could be manipulated to help treat disease. These control elements are regions of genomic DNA called enhancers that encode binding sites for transcription factors. While scientists have a decent understanding of how these regulatory regions work during normal cellular activity, what's less understood is how combinations of activators and repressors generate precise patterns of transcription during development. Today, we're going to hear how high-throughput methods have been developed to dissect transcriptional enhancers and understand their role in determining patterns of gene expression in a classic animal model system. But before we begin with the presentations, it's my pleasure to introduce you to our presenters for today's webinar. First up, we'll have a brief presentation from Dr. Maoyan Win, who is a Senior Technical Applications Specialist at GenScript. Win will tell us a bit more about GenScript and some of the interesting platform technologies that many of you may find extremely helpful in your current research endeavors. He will then turn the presentation over to our main presenter, Dr. Justin Crocker. Justin is a group leader at EMBL Heidelberg, where his lab focuses on understanding the fundamental principles driving development to allow for programming and predictive control of cell fates. Today, Justin will take us deeper into the role of enhancer architecture on gene expression by creating synthetic networks for pattern formation. But before we begin, I want to remind the audience to stick around for the Q&A session that will occur right after the last presentation. To ask a question, it's really very simple. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. You can do this anytime during the presentation, so don't be shy. Send in any question you might have. We're going to try to get to as many of your questions as possible. Okay, I think we're square now on how to ask questions, so hopefully many of you are feeling inquisitive today. And with that, I'm going to turn the presentation over to Win.
Hello, everyone. My name is Maoyan Win. I'm a Senior Technical Application Scientist at GenScript. Welcome to our webinar series. Today, we are very excited to have Dr. Justin Crocker to share some of his work in studying how transcriptional enhancers play a role in determining gene expression patterns and development in a model eukaryotic organism, the fruit fly. Dr. Crocker is a key opinion leader of GenScript, who has utilized our mutant libraries in some of his work that he is going to be sharing with all of us today. Before his talk, I would like to take a couple of minutes to introduce GenScript and some of our mutant libraries. For those of you who are not familiar with GenScript, we are the #1 provider of gene synthesis services in the world. We have 3,000 plus employees. Many of us are master's and PhD level scientists, so you can totally count on us. We have 30,000 plus citations in peer-reviewed journals with over 110,000 customers all over the world. We make DNA and more and help fuel advancements in various fields of biotechnology by serving as a contract research organization, providing innovative and high quality services to many researchers and scientists with our 3 core platforms: molecular biology, protein, and antibody. Under our molecular biology platform, we offer a number of mutant libraries for engineering a variety of proteins of interest, and the 3 most popular ones are shown here. Random mutagenesis libraries, as the name indicates, are for those who like to create mutant libraries randomly within a region of a protein sequence. This type of mutagenesis is often used when researchers have little information about the relationship between the sequence, structure, and function of a protein. Alanine scanning libraries are for those who are interested in identifying key amino acid residues responsible for a particular protein function, such as binding. And last but not least, precision mutant libraries are for those who have more information about their proteins of interest and are ready to take a more targeted approach in creating mutant libraries. For instance, the researchers probably already knew where to introduce mutations, as well as in many cases, they also have a set of specific amino acids that they would like to use instead of all 20 possible amino acids. With that kind of information available, we can help researchers to create smarter mutant libraries using only those specific amino acids. This kind of smart libraries can help reduce a huge screening burden on the researchers as they only contain desired mutants. For more information, please visit www.genscript.com. With that said, please welcome Dr. Justin Crocker for his talk titled Multidimensional Approach to Decoding the Mysteries of Animal Development.
I'd like to first thank the organizers for our opportunity to present our work, which is focused on decoding the mysteries of animal development. So really at the heart of the interest and a lot of the research within our group, we're particularly interested in understanding is this, how the simple linear arrangement of letters, these A's, T's, G's, and C's, give rise to these very beautiful patterns of gene expression you'll see during development. So what I'm actually showing you here is an early Drosophila development. This would be the developing head, this would be the tail. What I'm showing you here in these magenta stripes, each one of these is an individual nuclei. What you can see is you have this beautiful 7 stripes of expression that will appear reproducibly embryo to embryo and across evolutionary timescales that really shapes the development of these systems to ultimately give rise to the adults, fruit flies that many of us know very well. So what I really want to emphasize here is that these letters, which I'm showcasing in the back here, these are actually regions that are sufficient to drive the expressions of this particular stripe, this so-called second stripe of expression that we see for this particular gene. And the reason why we're so focused on understanding this is that from a gene regulatory perspective, which is what I'll be focusing my talk on today, we know an enormous amount about how these are actually working. So I'm going to walk you through an example of this here to kind of bring you up as an audience to a very similar point. So even skipped, or Eve, is a very, very classic developmental biology gene. And the reason we've been focused on it for so long within our group is because it's built upon decades of knowledge now. So what these particular regions, which we're focused on again today, these transcriptional enhancers, what they do is you can see that you have this 7 stripes of expression, and each one of these little gray dots here, these are actually a single nuclei. And what you can see then, the early embryo, you have these 7 stripes, and what's now known is that the individual regions shown upstream and downstream of this particular gene, these are actually sufficient to, if you put them in front of a reporter gene, such as GFP or any other fluorescent proteins, and you put them back into the fruit fly, these are actually sufficient then to drive an aspect of that gene's expression. Right, so what these switches, these transcriptional enhancers are doing, for instance, the second one right here, if you put this in front of the reporter gene, it will actually drive that aspect of expression of the second stripe. We see this also then for the stripes 4 and 6. Again, we have this transcriptional enhancer now, which will drive these patterns of expression. And you can see this for each one of these individual stripes. Right, so there's a very classic system, which has really taught us now how these genetic switches are actually encoding the information to drive these aspects of expression during animal development. What we want to emphasize then is that this is not unique for the fruit fly. And actually, you can see these invertebrate systems. And this is really another textbook example whereby you can see that you have the mouse embryo right here. And there's this gene known as Sonic the Hedgehog, where if you would take way upstream from this, there's actually an enhancer here, these transcriptional enhancers, which is a megabase away, right? So there's an enormous distance in the genome, and yet we know that it actually will contact that gene. It will thereby regulate the expression of the developing limb. And why this is so poignant for a lot of what we're talking about today is it's known that many mutations in transcriptional enhancers are actually responsible for human disease. And this is a very classic example where we're showing that if you actually remove this limb enhancer, in these mice, they will no longer develop functional limbs. And furthermore, in humans, if you have individual mutations to these regions, you can see that you have a very dramatic difference in actually the limbs and how they're developing. And really what I want to emphasize to underscore just how important these enhancers are to development, not just in flies and humans and everything else, is that in humans in particular, over 90% of human pathologies or medical conditions will actually map to regions which we posit are transcriptional enhancers, right? So they're very, very important, not just for normal development and for disease, but one of the other particular things our group is focused on understanding is how they shape animal evolution. And this is actually that same enhancer I showed you in the previous version, where this is a very nice example now to focus on what you can see if you look at mouse and compare it to pythons or cobras, which very clearly do not have limbs. You can actually track these down to individual changes then within these transcriptional enhancers of both these different families of snakes. And what you can do now within the mouse is you can actually introduce these same mutations that are found only in that transcriptional enhancer, and you develop, compared to the normal mouse, which has nice arms and legs, these now develop these severely truncated limbs, right? So again, this is just underscoring the importance of transcriptional enhancers for not just development, disease, but also towards evolution. And there's many, many just beautiful examples of how these are contributed. We think, for instance, for brain evolution, limb evolution, such as seeing a length, different digits between the chimp and the human. We think they have these very dramatic differences. For instance, if you look at fish and how these are actually relating to their ecology and how they're evolving in the wild to actually have these extra armor plates to escape predation. And finally, you see many, many beautiful examples, in this case, returning to the fruit flies, where you see how they're driving these pigmentation patterns in the very beautiful wings of these different fruit flies you can see in the wild. So again, changes in enhancers which are contributing and driving a lot of animal phenotypical evolution. So one of the things we're particularly focused on then within the group is, again, I'm going to return back to this very classic even skip strike 2 enhancer. And the reason we're so fixated on this is that this is one of the first transcriptional enhancers really that was identified and studied in depth. And what I want to emphasize then, if I was going to take this single sequence right here and present it to most any member in the audience and ask you to predict based only on the sequence, 1, is it a functional transcriptional enhancer? And 2, where is it driving expression during development? Most people in the audience would never be able to actually connect that to these types of expression patterns, such as the second stripe of expression we're showing here. So really one of the things that we're doing within our particular research group then is underscored here by this very classic, highly referenced quote by Richard Feynman, which is that what I cannot create, I do not understand, right? And this is a rather powerful heuristic, I think, for what we're trying to do within our particular research group, which is that if we really understand how these transcriptional enhancers work by first principles, we should be able to build them, which drives some predictive aspect of expression. And now I'm going to really walk you through an example showcasing how little we feel that we truly understand how these are working during development. And again, this is the quintessential textbook, literal textbook example, which I'm showing you right here, right? So this is a picture taken directly out of very classic textbook, which many of you probably read in the audience, which is Molecular Biology of the Cell. And if you were actually to Google right now, developmental enhancer, you could actually find images just like this one right here, which again, this is focused then on that second stripe of even skipped expression. This is very classic work by Mike Levine and Steve Small here. And really what's known now from a genetic relationship is the way you get very discrete stripes of expression shown here in this darker staining is that because what this is reading out is these inputs, which are hunchback and bicoid, which are transcriptional activators, right? So they're localized in these broad swaths throughout the anterior of the developing embryo, the head region. And what actually limits the expression, so you get a stripe, is you have repressors, which actually limit the amount of expression in these particular regions. So these are known as giant and cruple for the repressors, and the identity of the activators is hunchback and bicoid. And while it's not really relevant directly for this talk today, what I really want to emphasize then is genetically, a lot of groups have been focused on identifying these different inputs, and now we can actually map them all the way out biochemically to understand how they're binding to this piece of DNA, and really how then this binding relates to these gene expression outputs. So again, this is that even skip second stripe of expression. It's very, very precise in developing embryo. And if you now focus on that individual cis-regulatory module, right, that switch, that developmental switch which gives rise to this pattern of expression, the biochemical assays now have really footprinted what we think are collectively most of the information, at least that is identified up to this point, right? And these, you can see a variety of these variable affinities for those activators, bicoid and hunchback. And as well, we have different repressors, cruple and giant. And it's thought then that a very simple interpretation of how these are working is that you just have a combination of activators and repressors. And what these will actually in turn do is give rise to the very discrete gene expression patterns that we see. So a very simple example, if again, going back to first principles, if this is truly the information that's sufficient to drive these stripes, what we should be able to do is take these same motifs, scramble them randomly, and drive some aspect of expression. And really what you see here, which is I still think a very dramatic example, when you do this exact experiment, you take this enhancer, scramble the motifs, put it back in the fly, you see that you really have no expression in the developing embryo. You could say maybe something about the particular arrangement wasn't correct. You can scramble this again, and you see no expression. You can scramble this again, and still no expression. And you can actually repeat this over and over and over, and maybe occasionally get some faint aspects of expression in the anterior. You will never get that very precise pattern of this wild type stripe 2 expression you see in normal embryos. So clearly there's a number of different informations that we do not understand here. And one possibility in this case, you could argue then, is that maybe the arrangement, the spacing between these individual binding sites is actually important for this expression. So in this case now you can randomize all the intervening sequence, and what I want to emphasize is again here, you have no expression. Right, so clearly, this is one of the most well-studied, well-described enhancers of all developmental biology, and yet we still cannot build synthetic modules that will drive any expression. And I want to emphasize this is not just work from my group. Many different labs throughout the world have actually tried very similar experiments, and in general, we have nothing within the developing embryo that recapitulates that native stripe of expression. So really what our group is focused on now is exploring new methods and techniques to really go through and really at much larger scale understand what do we not understand about these particular regions. And this is what I'm really going to walk you through now as an audience. And in this case, what I'm going to focus on now is a minimal enhancer, not in the really, really early developing embryo, rather a little bit later. And I'll come around to why we think this is very important. But so again, this is another approximately 300 base pair piece of DNA. If we put this in front of a reporter gene and put it back in the fly, you again get these very beautiful stripes of expression, right? This is something as a research group we really are big fans of because it allows us to really go through and, as you'll see, quantify in a very discrete ways the levels of expression and how changes in the sequence might relate to that. So what we're interested in exploring now is focused on many, many questions which we realized very early on we do not have within our field a really solid understanding of the really basic biology of how these switches are working. And these questions are things such as how many binding sites are there, right? How do these transcription factors, how many are actually necessary for driving this expression? How are they arranged in these regions? How robust are these, right? If you start mutating individual binding sites, how does it relate to changes of expression? How evolvable are these? In other words, if there's this constant tension then between something very robust, it might not evolve that easily. And finally, can we predict patterns of change? Right, a lot of what our group is trying to do is actually to generate very predictive capabilities to understand how we can relate changes in these enhancer sequence space to these very discrete outputs we see during development. And the reason now we're focused on this particular enhancer, as opposed to something like the even skip, which I started the talk with, is that on the input side, so the genetic basis of this, we know an enormous amount of information. And the reason for this is actually highlighted right here in what's known as a very classic Heidelberg screen, which is a screen that won Yanni and Eric Wieshaus both the Nobel Prize. And the reason why this is such an important screen in biological terms is that what they managed to do is they mapped a huge number of almost all that we now know as these classic signaling effector pathways, such as Hox genes, wingless, EGF signaling, Notch signaling. And what's really important for what I'll be talking about today then is what Yanni and Eric were actually focused on is they were looking at these structures here, right? So these are not embryos, rather these are larvae, right? Or otherwise known as maggots in more common speak, right? So the reason that Yanni and Eric were focused on these is what you can see here. This is a wild type one, and you can actually see you have these very nice bands, right? What these bands are actually known as identical bands. And these are actually these little hook-like structures that the larvae will actually use to crawl along their substrate. And what's important here is what they actually did then is a very large screen where they mutated randomly the entire genome of the fly, and they found different examples like this where there were these very clear mutations, when genetically what they could do is figure out the signaling pathways by looking only at the denticle bands. What's important for our talk then, and my body of work, is that the gene that's known to integrate all this information to give rise to these very discrete structures is known as shaven baby. And one of these enhancers, if you take this enhancer and put it back into the fly in the reporter gene context, will drive these very nice aspects of expression. And again, what's important for us is that you see you have these 8 identical bands throughout the larvae, and these actually correspond exactly to the output of this particular enhancer, where you have these 8 different stripes of expression. So these are actually the nuclei in the early developing embryo that will give rise to these hook-like structures. So we have this very beautiful input-output relationship then, where we can start relating these classic genetic signaling pathways, how they integrate this enhancer, and ultimately how they give rise to these very precise patterns of gene expression. So the very unique approach which our group is focused on exploring then is a very large scale, very unbiased, and I think that's really key to what we're trying to explore, where the goal for this originally was to take an enhancer library with a random number of permutations in this. So in this case, we chose about a 2% fixed mutation rate. And really the reason we focus on the 2% is it's known that there's a very similar distance in terms of DNA sequence turnover that you get between wild and natural, or different natural flies in these closely related species. And we'll kind of return to some of this as we come towards the tail end of our talk. And so what we actually ended up doing then for this particular pipeline, which I'll walk you through now, is GenScript, who's been very good collaborators with us for the past, I'd say at this point, almost a decade, is that they built us this very nice enhancer library of a very nice fixed mutation frequency around 2%. And then what we did here is we created about 1,000 independent lines of flies. And then I'm going to walk you through how we built this very high throughput embryology techniques, which is the really centerpiece of this, is a lot of the robotics that allows us to handle all of this in a very automated fashion. And coupled with then the image analysis pipelines, that allow us then to relate changes in DNA sequence, right? So these very simple genotypic inputs to these very discrete outputs that we can see during development. So here in a little more depth then is the initial robotics, which we initially this started was in David Stern's group, which is at Genelia with the help of some very talented engineers, Jeff Jordan and Peter Polidoro here. And what we ended up doing then to build and push this technology, because we had 1,000 lines, which is a huge amount of embryos to handle. And on top of that, we really were going after very sophisticated assays with high precision and accuracy. And what I'm showcasing here then is the actual robotics is that each one of these fly cages you can see here would end up taking an individual fly line. And what these flies would do is lay a lot of embryos in these individual wells. And you can see an example of here where each one of these would be an individual embryo. So these wells then would directly interface with this robotics. We can hit go, and it will do all the fixations and stainings and really do everything for us, getting us ready for imaging. And again, what's really powerful about this then is it removes a lot of what the human has to do, right? So then if you have these kind of design, build, test phases, which is very common in synthetic biology, this allows you now to really increase the scale, the developmental context, to really explore how these are working during development. So the first assay to really get a handle on how this information is distributed with these enhancers is outlined here, and I'm going to walk you through it. And I really want to emphasize that this is the work of Timothy Fuqua and also Marlise, who is a very talented graduate student in the group. And what they ended up doing is first a very initial beta-galactosidase screen using the robotics just to see then how, again, these different mutations present in our library relate to expression. And the reason we use kind of a blunt beta-galactosidase enzymatic screen initially is one of the things that we found very, very quickly, which at the time was very exciting and kind of confusing, was that many of our lines, and this is a nice example of one that still has nice striped expression you can see, but if you compare that, for instance, this line 82-3, which is just present in our library, you can see that they have no expression, right? There's no expression present within these particular lines. And what we found is that this was not a unique example here. Rather, what I'm actually quantifying here is the percent or the number of lines with no expression as you increase the number of mutations. So what you can see is you go from 2 to 3 to 4, all the way up to 10, 11, etc. You can see you get this nice, smooth increase on the number of lines with no expression at all as you increase these number of mutations. So what this gave us then was a very large collection of many different lines with random mutations sprinkled throughout the enhancer, coupled with the expressions. And what we ended up doing then, a very simple scoring scheme saying, does it have anything that resembles wild type expression, or does it have no expression, such as these examples here? And taking all of this, what we could actually do is create what we call a footprinting score, where very simply what this is if you scare through all the mutations present in our scored lines, in this case about 200, or it is 274, you can see what this allowed us to do then was calculate a footprint score, which is simply the probability that mutating an individual site there would lead to no expression, such as right here. And what you could see very quickly, which agreed with our initial assessment of the number of lines with no expression, was that these are very dense elements, meaning that many regions present throughout this enhancer, if you mutate it, had a high probability of no expression. And what was quite exciting at the time is that many of these could actually map to motifs that were either consistent with prior genetic information, or these are ones that we have previously identified and validated within our groups, such as or UBX, which I'll go into detail a little bit further, or UBX. So the important point here again is that these are incredibly information dense, and we think this is actually underappreciated within the field. And we can actually explore this in other ways, such as what I'm showing you here. And this is simply much like a GWAS study. It's just the relationship between the probability if we mutate a site here, calculated a different way, leading to no expression. And again, no matter how you start to score these particular regions, you see that this is an incredibly information dense element. So at that point, these are beta-galactosidase assays, which are rather blunt for many of you in the audience may know. So we're focused now on actually increasing the resolution of these assays through much more standard microscopy methods. So what I'm outlining here now is the bit of our pipeline, which is focused now on taking the robotic embryos. We can mount multi-well plates where each individual well right here would be a different line. And you can see now if you look at an individual well, you can go in here, and what this actually is that each one of these little things that look much like a grain of rice is an individual embryo. Right, so there's a huge amount of data now where we can start to relating then how change of enhancers would actually at much higher scale, which I'm showing you here, relate to change of gene expression. And the reason we're really excited about this again is here we have different stage of embryos, so we can relate to timing events. And what we have now is very high resolution, very beautiful images, where now what I'm showing in this 3D rotating embryo. And I want to emphasize again, we can get this quality of data for every single embryo across all of these individual wells. You can see that what I'm showing now in green, this is the full composite of all that expression of this gene known as shaven baby. And what I'm showing you here now in magenta, this is that transcriptional enhancer which we've been assaying throughout this pipeline. So again, each one of these little particular regions here are individual nuclei. So we have just a wealth of information now throughout all these approximate 1,000 lines that we've been mutating. So the real take home of this right away is that initially what we thought we would go in, we'd have very simple assays where we could bin these, such as you'd have wild type expression. Maybe we had individual lines which have state changes or the probability of expression. Maybe we could have others where if you mutate particular regions, you might have levels, different levels of expression or the amplitude of expression. And then in others, you might relate that to different changes of location. What we found very quickly, and this was still incredibly jarring for us at the time, is that all, and I want to emphasize this, all mutations change expression of timing, levels, or location. right? So again, this feeds back to our findings that these are very, very information dense. And another important point, which I'll really kind of walk you through throughout much of our talk now, is that many of these are actually covariant traits. And what this means is that, it's just nicely showcased here, where you can see that these normal stripes of expression, where in wild type, they're very high levels, very smooth, continuous expression. Here you can see by mutating these particular regions, you actually decrease expression where you're normally expressed, but in this case, it's coupled now to extra expression throughout the embryo, right? We found this is a very common motif within our data set, is that while initially we thought we'd be able to put these in these separate bins, really we couldn't do this, right? These are integrated characteristics that would co-vary. And I'm going to return again to this throughout the talk. So first then, to really showcase just how sensitive this enhancer is to change, these are examples right here. Each one of these are single mutations with this enhancer. And you can see basically the qualitative expression here across different lines with a single mutation of this enhancer, as well as the quantification of this. We're comparing now to wild type, the fold change of expression. You can again see that every single one of these unique lines, you have a statistically significant difference of expression. And now we're interested then in relating it more in the evolutionary context to see if we could understand the relationship between how conserved an individual site is, which is oftentimes thought to be a signal for how important that site is in terms of the biology, and how much when we mutate these individual regions, how much would it actually change expression, right? And what you could see very quickly for us was that some of these would actually make sense. And what we have here is a Philo-P conservation score. This is just a metric for how conserved a site is. And you can see that some of these sites really might be strongly conserved, right? So presumably it's very important. And consistent with that, if you mutate this particular region, you can see that expression will go down very far. However, there are some that are much more perplexing for us, such as this is individual region here, this line 60-3, where you can see this mutation is actually not in the sequences that's well conserved. And yet, when we look at its change of expression, you can see it dramatically decreases. And we could actually extend this across the entire data set, where we look at the conservation score, relate that to change of expression, and what we found is there's really no clear correlation between mutational effects and conservation. So before we go too far into that, we want to see first then, could we validate some of these sites? And we focused here in this case on this particular region, because here it was kind of perplexing because we didn't have high levels of conservation, and yet this is one of the top ranked sites for our footprinting assays. When you mutate it, you again, just like this, you dramatically drop expression. So what I'm going to walk you through now is more validation at individual site. So this is an example of just the coverage we have in our library with the number of mutations per site. And what I'm showing you now are individual lines that we would pull out, which simply have a C mutation across this region that we're kind of zooming in and focusing on here. And now we're relating to that C mutation to how it actually changes the activity of the enhancer. And what you can see very, very quickly, again, is very, very dense information. And yet what you can start to do here is to footprint these individual sites, right? So if you mutate residues within this, you can see you dramatically decrease expression. And you can see a few of these that have a similar trend. And why we're quite excited about this is one of these we'd previously well characterized, what is known as a binding site for ultrabithorax or UBX. So the identity of this particular one is not that important to remember, just that we know now that it's a unique activator. When you mutate this particular region, you get these very dramatic decreases, right? So now I'm showing a very discrete perturbation to this region. If you compare this to wild type, you have a very dramatic decrease of expression. And finally, we can start to analyze what these other newly identified motifs are. And what was nice about this is they actually matched a consensus motif for a gene known as homothorax, which is known biochemically to interact with UBX. And what you can see when you mutate these, again, you have this very dramatic decrease of expression. So finally now, actually what this alluded to is that there might be some cooperative biochemical interactions between UBX and homothorax. And indeed, if you mutate both of these, you really have no expression in the growing embryo. So finally, I don't have time to talk about all this, but we biochemically validated all of these and their interactions for these homothorax sites, these UBX sites. We've identified additionally non-cooperative interactions for these homothorax, which I'm showcasing here, where if you actually go in, we managed to footprint these binding sites. if you mutate these two sites, you can actually see that again, you have a quantitative difference of expression. So the important point, what I want to really emphasize then, is we have a large, very dense amount of information. Many of these map to canonical motifs or some motifs that are much lower affinity. But these are the ones you biochemically validate in the type of assays I'm showing you right here. And in many of these, we've actually previously identified as being valid, important binding sites. So the next question I really want to focus on then is why are they not conserved, right? This very classic metric we thought where it's very important, it should be constrained over time. So for this, what we focused on then is these two different Drosophilids, which have been diverging in this case about 40 million years. And what you can see that melanogaster, if you compare it to Virilis, Virilis is this very large, much larger fruit fly. And if you actually do a conservation, if you look at the sequence conservation here in these type of plots and look at the identity, you can see that it's really variable across this particular enhancer. And what you can do then is actually break it apart in some of these different motifs we've been focusing on. And what we have right here, this is that 60-3 line, or that homothorax line. When you delete this, you have a very dramatic decrease of expression, right? Something just like this, where again, this is an example where if you compare it to wild type, by mutating this region, which has this variable conservation, right? It's not that well conserved. You can see you have no expression. And this is contrast to other sites as UBX here, which is deeply conserved across all of these different Drosophilids. So we're interested then in why the Virilis enhancer really was not that functional. So to simply test it, which I'm showing you here, is that when you put the same enhancer back into melanogaster and start to quantify the levels of expression, you can see that they're actually indecipherable from the melanogaster variant where we simply delete the site. All right, so when you actually focus again on this enhancer to really hammer this home, is that this particular residue, we cannot tell really functionally how it's different than by mutating it from melanogaster. And what's quite compelling about this now is we can add this homothorax site back into the Virilis enhancer, and we can actually kind of rescue the levels of expression. So it goes up almost to the levels of wild type. And finally, why this is quite compelling for us is now if you actually go into the trichomes, right? So this is a zoomed in version of what I'm trying to showcase here on these denticle bands, which I mentioned earlier. You can see that melanogaster has a smooth field of these little hook-like structures, which again, they use to help crawl. Virilis, which is entirely consistent with this data I've outlined here, actually is missing these little hairs, which are exactly where this enhancer is driving expression. Right, so we think potentially as we start to learn more and more information about these enhancers through these type of high throughput approaches, it's going to allow us to have some predictive capability to really going back out into the wild and understanding how these systems might have evolved. So at this point then, to kind of take pause to really see how much I've been demonstrating towards you as an audience, is that we think that this enhancer is very information dense. We think cooperativity makes this enhancer incredibly sensitive to SNPs. And we think part of this, which I'm going to walk you through for the remainder of the talk, is this allows us a new data set to explore how evolvable are developmental enhancers, right? And this is a very big, compelling question within the field of evolutionary biology, which I'm going to kind of walk you through and break down at this point. And the key take home here is this concept known as developmental constraints. And so here's a very good quote, which I'm going to walk you through right now by reading it out loud, which is developmental constraints are thought to be the property of a trait that, although possibly the environment in which it originally evolved, acts to place limits on the production of new phenotypic variants. And historically, there's a very similar concept to what Darwin, the father of evolution, or one of the fathers, would call unity of type, in which what they found is that there's certain traits they thought that might not independently evolve, rather there's a trend or a directionality of how these different traits will evolve. And in this case, what you can see in a more modern style assay, what they're relating to in the form of beaks is the brain case size, how it's actually interfacing the skull and the beak length. And what they found is to actually sample ecologically with different bird species is that there's, again, this very classic signature of developmental constraints, which is that these traits will co-vary. So what we are interested then is to see, does our enhancer logic, right, was this logic place limits on the production of new phenotypic variants? So now we're actually returned back to our original enhancer assays and start exploring how our library contains information that could give us really a clue to explore this relationship. So we have here is the wild type enhancer, E3N, and you can see normally in the anterior, you have no expression. It's very quiet transcriptionally. You can see your normal aspects of expression, where you have these nice stripes, because in between these stripes, it's known that these are actually being repressed, right? So it's being shut off in between stripes. And we knew previously that if you actually increase the affinity for these activators, presumably so it binds with a much higher affinity, what this could actually start to do is you have much higher levels of expression where you're normally expressed. You start overcoming that expression in between the bands. And on top of that, if you increase the affinity, what starts to happen is different genes can start to bind there, right, or different proteins. And what this does is where you're not, UBX is not normally expressed there, right, this high affinity variant. This activator input is not expressed there. Rather, other things are going to start to bind there and turn it on. And what's quite powerful about the type of assays and the very high throughput approach is that normally what we have, we've been focused on analyzing things in a much later stage of development, in this case, stage 15, for people who are FLY audience members. And if you look a stage earlier, you don't normally see expression of this particular enhancer. However, by increasing the affinity of this particular variant, you can see that now you turn on in the embryo at a stage earlier. And this is just a simple quantification of that, where the normal wild type enhancer is not expressed in the anterior or really anywhere at the earlier stage. In contrast, shown here in green, if you compare this to wild type now, you can see that these type of variants, by increasing that affinity, you'll turn on ectopically within the embryo in these anterior regions, and you'll turn on earlier. And now by stage 15, if you compare these two, again, you have much higher levels of expression, which is coupled now with anterior expression, where you're not normally expressed, and these naked regions, which are normally being repressed in between these bands. And now what we can actually do, because we have a much larger number of these variants within our collection, we can now start relating these different trends and how they change across our libraries. So now I'm actually focused on normal aspects of expression of stage 15. We're looking at earlier expression or precocious expression, as well as the ectopic anterior expression. We're relating all these variables now to the affinity, the total affinity of UBX across that enhancer. And what you can see now, based on the size of these circles, you can see that you have these increasing affinity across these enhancers for UBX. And what in all cases this correlates with then is these linked relationships between the level of intensity, precocious expression, and kind of overcoming that repression in between segments, right? So again, these are highly linked covariant traits, which is a signature of constraint. And while I don't have time to go into this, if anybody really wants to go through further depth about this relationship between kind of specificity and affinity, I'll refer you to my previous work, which is the reason we think you have this ectopic expression is you increase affinity, you're going to have kind of cross competition between members of this large family of proteins. And the next thing we're really interested in understanding then is really what was possible with the enhancers. And in this case now, we're kind of taking a prior information we had where we knew that it was theoretically possible by changing the transcriptional inputs in a very blunt way, in this case by a wingless signaling mutant, or this is TCF signaling in vertebrates. We knew that if you compare to wild type, again, you have these nice clean denticle bands. By completely removing this signaling effector, you get this lawn of trichomes, right? This very, very dramatic phenotype. And what's known about this particular signaling effector is that the way it's driving the expression to repress genes is that normally you have these hook bands within the embryo, of which E3N is expressed. And in between this, you have this wingless signaling, which actually, through these TCF motifs, will repress expression there, right? So if you remove wingless completely, you have this line of trichomes throughout the developing embryo. And now what you can see here, if you look at the logic of the enhancer, is that in most cases, these TCF motifs will be proximal or directly overlap one of these activator sites, such as that homothorax site I walked you through so much earlier. So what we did very quickly is we could look at our libraries now, pull out variants of these individual enhancers, which are unique to these TCF motifs. And this is an example of a single SNP. And you can see that TCF will overlap partially that homothorax motif, which is critical for expression. And now if you mutate this single nucleotide in that TCF site, you can see that your normal aspects of expression in this stripe will actually decrease slightly. And this then is coupled with an increase of expression now in between these bands where it's normally repressed. And here's a quantification of that, where you see that wild type is expressed at very high levels in the normal zones with very, very low levels in between the segments. And now if you start looking at different variants, you can see this very clear trend that starts to emerge, which is by mutating TCF sites, your normal levels of expression will decrease, but this is always coupled then with an increase of expression in between these regions. And we've actually extended this over to over 20 different enhancer lines now, and this trend is very, very common and holds very, very well. And you can actually map this out here now, and again, the kind of this broader space in which now you can start to relate your normal stripe intensity, whereas you start to mutate it, you'll decrease expression. But this is actually then coupled with some anterior expression, where the expression gets a little noisy and starts turning on where it's not normally expressed. And this is actually then always coupled with the expression in between the segments, right? So you have, again, this very, very linked, apologize for that, you have this very linked changes of expression across the enhancer. And then a very, very clear test of this then, because we thought there's truly this constraint operating on the enhancer, is if we actually go out into the wild, can we see, is this actually predictive? So we actually tested now is over 50 different Drosophila larvae across all these different species. And what I'm highlighting here, and again, I want to emphasize this is over about 150 million years of evolution, and cumulatively this is almost 1 billion years of evolution. You can see that if you look across all these different larvae, we have not a single example of a gain of expression in between segments, right? So again, we have multiple examples now showcasing here in red of particular lines which it might have lost a particular aspect of this expression. And this, in this case, is a particular line which is, you can relate back to the sequence space, where this has lost a lot of enhancer information, right? So we really think then this is providing us some predictive capability, because if we start to understand the systems, we understand how they might evolve and how that might change based on the logic of these transcriptional enhancers. By understanding the relationships, understanding these constraints, we think it's giving us a predictive capability. And again, just to really highlight, this is a very classic book where we think now what these large-scale synthetic approaches are going to start to provide us is what Francois called these adaptively coherent patterns, right? And by exploring not what we actually have through either standard variation or what's constrained, we can explore a much larger space of what's possible, right? So again, this is what our large collection of enhancers are starting to provide us, is what possible relationships could have emerged if we actually decouple this then from fitness. So to really just take pause then to kind of wrap this talk up before questions, what we want to emphasize that is we think these minimal enhancers are extremely information dense, right? And this has really been underappreciated, we think, within the field. And going all the way back to the start of the talk, we think this is why it's been so hard for us as a community to actually be able to identify these elements and actually predict what transcriptional enhancers are doing, and to actually be able to engineer or build them. We think that there's extensive pleiotropy, meaning that when you mutate one little enhancer region here, it has this extensive knock-on effect where it's going to start turning on in different tissues, which I walked you through with both the TCF mutations, the repressor mutations there, or by increasing affinities. And we think really, and we think this is a very important point because it's for the field, for the evolutionary developmental biologists, as well as people looking for predictive capabilities, that the enhancer structure is producing developmental constraints. And I really want to thank then GenScript, who's been just really instrumental in helping us as a group, just building and exploring biology in a very, very new way, right? Because this is really affording us now just these really new approaches to go in at scale to really change and modify and really to understand biology, developmental biology, we think in a very new way. Really have to thank you for this relationship. And we think, again, this is affording us a way to really exploring what's possible in the genomes, to really understand from a very new angle, a new perspective, how these are actually encoding as genetic information. And with that, I just want to thank all these members of the lab, everybody who's really contributed over the years, as well as David Stern, who really contributed heavily when I was at HHMI in Genelia Research Campus. With that, I'll take any questions. Thank you.
Justin, great presentation with some really nice insights into how gene expression patterns are controlled during animal development and disease. So thanks very much for sharing that with us. To the audience, stick around. The Q&A is coming up in just a moment. To ask a question for Justin, it's really simple. Again, all you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. All right, since we have a bunch of questions lined up already, let's get to the Q&A session and try to get through as many questions as possible. Bear with us for just a moment as we transfer into the Q&A session.
All right, everyone, thanks for joining the Q&A. We have a bunch of really great questions for Justin, so let's try to get to them. Justin, one of our audience members would like to know, to what extent does the distance between the TF binding site to allow combinatorial binding, chromatin opening, and DNA structure play a role in maintaining enhancer functionality?
Yes, this is a great question. The short answer is each of these is very important for enhancer activity. But a slightly longer answer then is that we know that many of these will interact with each other when they're bound to DNA, so such that they can have synergistic effects to enhance binding. And chromatin opening is also very, very important in the sense that there's some factors that will bind there and actually will open up the DNA and then be permissive for other factors to bind. And finally, the third part was, I believe, the structure of DNA. And again, this is very important because we know, for instance, you're going to have the shape of DNA, which can dictate the binding kinetics of how factors will interact with that. So yes, again, each of these are very, very important.
All right, thanks for that. Next question. Why would an enhancer be a megabase from the gene? Is this an exception or the general rule?
Oh, the why part is tricky, but this is, in this case, is a very extreme example. Typically, they're closer to the promoter, which would drive the expression. There's often kind of the probability is much higher that's close to the promoter and tails off. But in this case, it's thought that some of these might control the rates, meaning that if it's very far away, the probability of interaction is lower. So this is actually used, for instance, in olfaction to help kind of steer the system in a very clean way to control different aspects of gene expression and to actually, in this case, dampen it. It's a good question again.
All right. Thanks for that. Next question, an audience member would like to know, what did you use to locate your enhancers to start with? Did you use the Finish Enhancer search software, or did you use something else? Or was it just well known already?
Yeah, so this is actually, so this enhancer was previously deeply characterized by some of the groups I've worked with. And this was through just sheer force of will and doing a lot of what we would call enhancer bashing. So just systematically carving across, 100 kb of this locus to see which would drive expression. In modern times, a lot of what we're doing are more ATAC-seq or DNA-seq based ones to look at regions which are actually open and have different histone marks, for instance. So I'm not familiar with the Finish Enhancer search software, but if anybody ever has any software you think could be leveraged towards what we're interested in understanding, I'd be more than happy to try it.
All right, thank you, Justin. Next question, one of our audience members asks, for your enhancers that appear not to be conserved, have you looked at redundancy in DNA binding motifs, i.e. could binding be conserved, but sequence is not?
Oh, beautiful question. Yes. Yeah, so this is something that we're very actively exploring right now. We've published a little on this previously, but what we do see is that some of these will actually move around within the enhancer across evolutionary timescales, which is known as, for an evolutionary biologist, stabilizing selection. So in this case, the sequence would not be conserved, but we think the total affinity across the enhancer for many of these motifs is conserved.
All right, and looks like we have one more question here. Very poignant question. Do your robots and imaging techniques work for other systems?
Yeah. Well, yes. Is another short answer. A lot of what we're doing right now is interfacing with others who want to adapt some of our robotics and our approaches. And then again, this is, if anybody's interested, feel free to ping us. We try to attempt to make as much of this as open as we possibly can to get out to the community. Because a lot of what we're trying to do is kind of very basic bench scientists, is to really I think, raise the standards of the field and make it a very nice entry point so people can reproduce a lot of the experiments. And again, kind of really do science in a new way. Because a lot of what the technology is enabling us to do is to really ask new questions. And really, as we kind of walk through today, to really address these in depth and at the scale, which is going to give us, we think, the resolution that requires to understand how the biology is actually playing out within these very, very complex systems. So yes, if anybody's interested, we're more than happy to share any techniques, technology, etc.
All right, thank you very much, Justin. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the Gen website at genenginews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any friends or colleagues you think might be interested. I'd like to thank Justin and Wynn for their informative presentations, and I'd like to thank the audience for their attention and very thoughtful questions. And a very special thanks to GenScript for sponsoring this webinar. Hopefully we'll see you again in another Gen webinar in the near future. Everyone stay safe and healthy, and goodbye for now.